Okay, so let me continue. So today we'll be studying the Rhizopus, right? And I'm sure most of us have seen Rhizopus before. I hope we've all seen let's say carbohydrate foods that are going still, like banku, fufu, or even bread that is going stale. I think we've all seen these foods before, and we've all noted some quite fluffy substances that grow on top of them. If we have, please unmute and tell me you have, or you can give me a brief description of how they look like. Have we, have we all seen this before? So you're asking on top of what I'm saying. If you've noticed some carbohydrate foods, you notice that when they are going bad or when they are going stale, they develop some quite fluffy mass on top of them. Like you have bread or bamboo or even food. Sometimes it grows on the surface. You have these quiet substances that shows that the food is getting spoiled. I hope you've all noted that before. That's what I'm talking about.
Yes.
Okay, so that's fluffy substance is what we are going to be discussing today, and it's known as the Rhizopus, right? There are a lot of species of Rhizopus. There you can have Rhizopus oryzae and other forms, but in general, we are going to be talking about the Rhizopus. And what we need to know about the Rhizopus would be classification, reproduction, nutrition, and then economic importance. Those are the most important things. Because it's a fungi and it grows on a substrate, that doesn't necessarily move, and then respiration will be by simple diffusion and all that, since we know of these microorganisms. So there will be no need to delve into details of the other life processes it undergoes. So the most important part to be taking a look at will be the body morphology and nutrition, reproduction, and then the economic importance. Is that okay? Okay. So I hope you can all see my screen. I'm sharing my screen. I hope you can all see that. Oh, you're asking if, yes, that can be mold. You can also have Rhizopus growing on top as well, looking like a quiet, fluffy substance. Mold can sometimes be green in nature if you study. Okay, so can you all see my screen?
Yes.
Okay, so as you can see on the screen, I'm saying that the Rhizopus belongs to a genus of saprophytic and parasitic fungi. So what you can know from the first statement is that the Rhizopus is a parasitic fungi. So parasitic meaning it feeds on other organisms or gets its source of food from other places. And then saprophytes. Saprophytes are organisms that feed on other organisms as well, but they digest the food outside the organism before they absorb it. So that's what saprophytes are. So these are forms of feeding. When you further down the course, you get to know the different types of feeding, saprophytism and all other stuff. So the Rhizopus belongs to a genus of saprophytic and parasitic fungi, and they are located in damp or moist environments. These can be found on organic foods such as fruits, vegetables, bread, etc. I've already said that. Now, looking at the body is, we say it's sinusitic. And what sinusitic means is multinucleated. So if you take one of the cell, you can have a lot of nuclei or nucleus within one particular cell. Do we get it? And then the body is highly branched, right? It's highly branched, and that's what helps it form the fluffy mass, which we call the mycelium. Please, I hope you are okay.
Yes.
Okay. So some of the Rhizopus species can infect humans with some form of diseases. So now looking at the classification, now Rhizopus, because they have a true nucleus, which is membrane bound and has membrane bound organelles, it's a eukaryote, hence it belongs to eukaryota. Now, as I said before, it's a fungi, so obviously belonging to kingdom fungi, phylum Zygomicrota, class Zygomycetes, order Mycorrhizae, family Mycorrhizae, and then genus Rhizopus, right? So its genus is Rhizopus, and that's what gives it its name. And if you delve deep into biology, when you do scientific naming, you realize that if you are naming organisms, you name them using the genus and species name. So that's why you can have the binomial nomenclature, right? So, for example, if you take one of the species of Rhizopus, such as Rhizopus oryzae, then you can say it belongs to the genus Rhizopus, and then its species name is what? Oryzae. Hope you are good. Okay, so let's continue. Now, the Rhizopus, as I said before, can be seen as a white fluffy mass that exists on the suitable substrates. And what are substrates? Substrates are, let me term them as organic substances that provide the necessary nutrients required for certain organisms to survive, right, and carry out their normal life processes. That's simply what we call a substrate. So if you find Rhizopus on bread, meaning the bread is providing the nutrients or the resources that are necessary for the survival of this Rhizopus. So the bread becomes a substrate, or the carbohydrates in the bread is the substrate for the Rhizopus. Hope that is clear. So on the normal, to the naked eye, you'll be seeing the Rhizopus as a white fluffy substance, but under close inspection with the microscope, you will find out that they are made up of thread-like structures, and these thread-like structures are known as hyphae, right? So you have different forms of hyphae. We'll be looking at that further down. So you realize that you see the fluffy mass, all right, like a ball of cotton. But if you view it under the microscope, they are made up of tiny threads, and these threads, as I said before, are synocytic multinucleated cells, right? So these thread-like structures are known as the hyphae. And then when you put a lot of hyphae together in a total, that's what forms the mycelium. Please, are we good?
Yeah.
All right. It looks like just one person responded. The others, are we okay?
Yes, please.
All right. So, as I said, the hyphae can exist in various forms. So you can have the horizontal form of the hyphae, which are known as the stolon, right? Please note the terms I've been using so far. I've mentioned synocytic, I've mentioned that it's parasitic, saprophytic, I've mentioned hyphae, I've mentioned mycelium, and now I'll mention the stolon, right? These are terms that will obviously pop up in your exam, or you'll be required to know. So the horizontal hyphae are known as the stolon. Now you can have root-like structures in the Rhizopus, and these will not be called roots here, but these are called rhizoids, right? And these rhizoids are most of the time just one cell thick. Let's take note of that. They are most of the time rhizoids are like one cell thick. And then the less erect structures that arise from the point where you have a stolon and then a rhizoid meeting. The structure that arises from that point is known as the sporangiophore. Let me show you the diagram to illustrate what I'm saying. So you have these parts. Please, I hope you can all see my cursor pointing to the screen.
Yes.
Okay. So you have these horizontal parts, right? These parts are known as a stolon. Then you have these root-like structures here, which I said we will not be calling roots, but these are what? Rhizoids. Okay. So you have the rhizoids here. Now, the point where you have a stolon and a rhizoid meeting, you have a structure coming out from that point. And that is what we are calling the sporangiophore. Are we good? Now, at the tips of these sporangiophores are something we call the sporangium, right? That's for single, and then plural would be sporangia, right? Okay. So that's about the morphology. Now, the sporangiophores bear round reproductive structures at the tips called sporangia. And this sporangia, most of the time, when they mature, they bear spores, right? So these spores are like, they play the roles of seeds, let me say it that way, but they are not seeds. Let's take note. So these are spores. And then at maturity, the sporangia develops these spores, forms the columella, and then when it's ripe enough and it finds a suitable substrate, it ruptures open, releasing the spores into the suitable substrate. So that's the whole thing about the Rhizopus. So what I'll need you to do is learn how to draw a Rhizopus and properly label it. It mostly comes up. So now, for roots, we know how the role they play, and then it's similar here for the Rhizopus as well. So here the rhizoids, they serve as a point of anchorage. So if you have these mycelia forming on a suitable substrate, obviously they need a support on the substrate. So you have these rhizoids playing that role. So they grow downwards, right? They grow downwards into the substrate. You later learn this form of movement. It's called the gravitropic movement, or this is a positive gravitropic movement, right? So you learn that later on. So it moves downwards into the substrate, anchors it, and then because it's one cell, it's able to exchange material or nutrients between itself and then its environment. Please, I hope you are good.
Yes.
Are we good? Any questions from where I've started to this point?
No.
Okay. So now, taking the hyphae has a wall, right? You can actually have the cross section of the hyphal wall, right? And you find a lot of things in there. So the hyphal wall is made up of chitin, right? You learn this. So most of the time, fungi are made up of chitin and then other substances. Other organisms and what their cell walls are made up of. Plant cell walls are made up of cellulose. You can have some bacteria made up of peptidoglycan and all that. So yes, let's note that. So because this is a fungus, it's made up of chitin, and the chitin is a mixture of some proteins and then complex sugars. Let's take it as that for now. So the hyphal wall is made up of that. Now the hyphae is tubular, meaning it's tubing, it's like a tubing shape for conduction, right? And non-septate, meaning it is not divided by cross walls. Do you get it? So you can have one hyphae. There is no breakage, like how you can have certain stems and then they have nodes in them. That doesn't happen with the hyphae. Do you get it? So it's tubular and non-septate. It's not divided by any cross walls. The cytoplasm is granular and contains cell-bound nuclei. What's the term given to the severally nucleated cell? I mentioned that at the beginning. What's the name given to a multinucleated cell? Anybody? Okay. So remember, I said it's a synocytic cell, right? So let's remember that. Okay. So now
Please come again.
Which part? You asked me to come again. Which part?
The word you just mentioned for a cell with
Synocytic.
multiple nuclei.
Synocytic.
Okay, thank you.
Yeah. It's right here.
Synocytic.
Look on the screen. I'm pointing at it. Can you see?
Okay.
Yeah. Okay.
Yes. Yes, please.
All right.
Thank you.
Welcome. Okay. So for nutrition, obviously, because it's a white fluffy mass, the first conclusion we can draw is that it lacks chlorophyll, or even if it has chlorophyll, the chlorophyll is masked by different pigments. That's the reason we don't see green, right? Because we know that most of the time the plant has chlorophyll, The obvious color is green. Hope you all know this fact, like from teachers. So yeah, we are seeing the Rhizopus as a white fluffy mass, so we can draw a conclusion, a hasty one, that it doesn't have chlorophyll. And if a plant doesn't have chlorophyll, obviously it's not going to produce its own food. So it needs to have certain mechanism that is going to help it acquire food from other sources. We get that. So now, looking at the Rhizopus, I mentioned the rhizoids, right? So now the Rhizopus wants to feed. It wants to feed, right? So what it does is it locates a suitable substrate. Then the rhizoids starts their action. So the rhizoids anchor themselves into the substrate and they start tapping into nutrients. So this is saprophytism, right? So because it lacks chlorophyll and the mechanism for it to produce its own food, the rhizoids do the work. So the rhizoids anchor themselves to the substrate and then begin tapping in the nutrients. Now, we all know that digestion needs to take place before the nutrients are absorbed. And here lies the case that the Rhizopus doesn't have this mechanism. So what it does is that enzymes are released from the rhizoids into the substrate. And then what happens is that these enzymes that are released break down whatever food substances you have in the substrate. So most of the time, because we said they are mostly found on carbohydrate foods, you have the enzymes breaking down simple sugars, right? You can have these enzymes breaking down the complex sugars you find in these carbohydrate foods. It's going to break them down into simple sugars. And then these soluble forms of the simple sugars are just transported either by simple diffusion into the cells, and then the cells transport it through the hyphae and then throughout the mycelium. So as I said before, organisms that mostly digest the food outside their body before they absorb the nutrients that arises from the breakdown are mostly saprophytes. And as you can see from the slide, all saprophytes do this because they lack the mechanism to digest the food substances. So they do so by releasing enzymes, right? These enzymes break down the food substances. And then it gets into the most simple and soluble form, which is absorbed by the rhizoid cells and then into the hyphae of the mycelium. Please, are we good? Any questions so far?
No.
Okay. So that is the mode of healing of the Rhizopus. Okay. So now moving on to the reproduction. So as you've seen, the other organisms that we study, the Rhizopus can also undergo sexual or asexual reproduction, right? And then the asexual reproduction mostly takes place in suitable conditions. So where conditions are suitable, it wouldn't like to waste resources because sexual reproduction involves a lot of energy, a lot of breakdown, a lot of buildup. So where conditions are suitable, why would I stress myself to undergo sexual reproduction when I can just undergo a simple asexual reproduction and then I'm good to go? Do we get that? So sexual reproduction will take place in unfavorable conditions where it needs to survive, it needs to maintain that stream, but yet the conditions are unfavorable. We get it. So now, the asexual reproduction is by sporulation, right? Sporulation and production of spores. So during the growth of the mycelium, the sporangiophore, as I said, the sporangiophore is the part that arises from the meeting point of a stolon and a rhizoid, the vertical part. So the tip of the sporangiophore keeps growing and then enlarges, right? It keeps getting enlarged. Now, as it gets enlarged, there are certain nuclei that will migrate within the hyphae from the sporangiophore into the tips, the enlarged tips, and then it gets cut off by a cellular wall and then forms the columella, right? Then the cytoplasms within the columella and the nuclei would then fuse together to form these spores, these little spores that we have. Do we get it? Let me take it again. So you have the sporangiophore growing, the tip of the sporangiophore growing and swelling or enlarging. Then as it gets enlarged, it gets filled with the migratory nuclei from the hyphae below to its edges, which is known as the periphery, as you can see from my slide. So then the central portion of that sporangiophore will get cut off by a wall, which will contain mini vacuoles and other cellular materials. And this part is what we call the columella, right? Then the cytoplasm with the nuclei will develop into spores. So now let me take you back to the picture, as you can see from here. So you can see this part here, which we are calling the columella, being cut off, is being cut off by this wall here, which is the central portion being cut off by this wall. Then you have this part, the peripheral cytoplasm, and the nuclei you have in there would develop into spores, as you can see. So we can say this is a mature sporangium. Why? Because it has spores in there. So we are talking about the asexual reproduction here. So at maturity, what happens is that the sporangia would absorb water from the atmosphere and break open. So it looks like dispersal of seeds, right? It looks like the dispersal of seeds. So it absorbs the water, swells, bursts open, releasing the spores. Now, it will only do this if the conditions are favorable, meaning it has a good substrate where if it releases the spores have a viable or a likely chance of germinating into a mycelium, a new mycelium. Do we get this? So at maturity, absorbs water, breaks open, releases the spores, and most of the time, because these are very tiny, they are transported by wind. So you can call it a form of wind dispersal. So the wind transports them, and then you can find these spores falling on suitable substrates. So you can have them in some flour, you can have them in certain food substances, such that because these food substances are suitable enough, when they fall there, they're able to germinate again and form a new mycelium. That's how come sometimes we wonder why you keep certain foods down. You feel like it's the most hygienic preservation you've done, but then it will still get spoiled and form some mycelium. Please, can you all hear me?
Yes, please.
Okay. So that's how the asexual reproduction occurs. Are we clear with the asexual reproduction? Please, are we all clear with the asexual reproduction?
Yes.
Okay.
Please, I can't hear you.
Can you all hear me? Those complaining can't hear me. I don't think I can do anything about it. Please check your internet or rejoin the class again to see, because the others can hear. So can I continue? Okay. So that is with the asexual reproduction. Let me see if I have a diagram within my slide to show you. Good. Good. So this is it. You have your vegetative hyphae. These are rhizoids. So you have your rhizoids forming in the suitable substrate, right? Then you have your sporangiophore developing. At maturity, there's the columella formation cut off by this wall, as you can see. Then it begins to form spores from the peripheral cytoplasm and then the nuclei. Because it's multinucleated, it's able to form these number of spores. We get it. So it keeps growing, keeps developing a lot of spores till it gets mature. When it's mature enough to release the spores, the sporangium would absorb water, swell, burst open, and release the spores that look like seeds. Because they are light in nature, they are carried by the wind onto suitable substrates. When the conditions are favorable enough, then they fall on the substrates, develop a germ tube into the substrates, and then the cycle begins again. So this is an image showing asexual reproduction in Rhizopus, right? Okay, so now let's move on to the asexual form. The sexual form of reproduction. So the sexual form of reproduction, as I said before, is for unfavorable conditions. So it helps it survive in adverse conditions such as drought and excess heat, right? So obviously sexual, so we need two parents, a male and a female, but here we call them the plus and minus strain. That's how we call them. So you have two hyphae, a positive or a plus hyphae and a minus hyphae.
Please, I can't hear.
Can you all hear me? Can you hear me, please?
Yes.
Okay. So I was saying that the sexual reproduction involves obviously two parents, right? So in this part, we call them the plus strain and the minus strain, right? So you have these two coming together in sexual reproduction. Now, for sexual reproduction to take place, you have a short stem being formed from the hyphae of these two. And this is what we call the progametangia. I'll show you a diagram shortly, but listen. You have these short stems being formed, which are called the progametangia. So they swell up and they keep moving towards each other. So you have the two hyphae side by side, then you have the progametangia being formed, moving together. Then as they meet, a crossword develops, and then the tip of these progametangia are cut off, and now they become gametangia. So as I said before, is before. So before they become gametangia, that's what happens. They keep growing together till they meet. The crossword forms, the tips are cut off. Then you have the gametangia being formed. Now this structure is being supported by what we call suspensors, right? This structure is being called supported by suspensors. Let me use the diagram to explain so you understand. So you have your strains. As I said before, you have the plus strain, the minus strain here. Then you have these structures being formed, which we are calling the progametangia. So you have the singular being progametangium. So you have that of the positive strain and that of the negative strain. They keep swelling till they get together. So now when they get here, a crossword is being formed. Tips are cut off, and now progametangia becomes the gametangium. Do we get it?
Yes.
Okay. So now the gametangium, let me go back to the slide. So now you have the gametangia being formed, supported by suspensors, right? Now you have the cellular material of these two mixing up, right? You have the contents mixing up, nuclear pair, will pair up and fuse and all that. Now, there's some form of, let's say, cross-fertilization or fertilization, kind of. Then you have a zygospore being formed. Now, when the zygospore is formed, it gets disconnected. So meaning these suspensors which held the gametangia together would shrink. Then the zygospore is resilient. So it's black, a thick-walled, right? And because it's thick, that is what helps it survive the harsh conditions. Now let's go back to the diagram. So you can see gametangium, there's the mixture of the cellular material. Then you form this hard structure here, which is known as the zygospore. Now, when the zygospore is ready to develop into a new hyphae, which will then go on to to form the mycelium. What happens is that these suspensors would shrink. They'll dry off or shrink off, leaving the zygospore alone. So now the zygospore remains dormant, right? So meaning it has no work to do until things or until it feels it's ready to produce a new hyphae or a new mycelium. We get it. And please, when the meeting ends, I would like us to join again with the same name because we have less than a minute for it. Okay. So now you have the zygospore becoming dormant, right? Awaiting further instructions for it to further divide and then form a new hyphae. So when it receives such instructions and things are good to go, what happens is that the zygospore would form would get on a new suitable substrate, right? This substrate to provide the necessary conditions, water and nutrients, suitable enough for the zygospore to burst open and then germinate. As it germinates, it produces a new hyphae. So this zygospore